Hello students, a warm welcome to all of you in ITLS Academy and also welcome to all in our 6-month online certification course on topic Artificial Intelligence and Molecular Biology. Now we discuss the next method, that is a genome sequencing. DNA sequencing. Okay, first we discuss the first generation DNA sequencing method, that is a Sanger sequencing method. Okay, by the help of this diagram we understand how we will perform the DNA sequencing, or basically Sanger DNA sequencing method. First we perform the denaturation process. Okay, first step is denaturation. Okay, second step is amplification. Okay, this step is performed in PCR, that is a polymerase chain reaction. Okay, then this is a denaturation, this is a multi-copy of the segment or amplification. Okay, then we take a 4 test tube. Okay, first test tube, second test tube, third test tube and fourth test tube, and we add this primer in this test tube. In this test tube, in first test tube, second test tube, third test tube and fourth test tube. In all test tubes we add DNA primer, DNA polymerase, primer, T and DPs. Okay, now here one point is very important. This point is, suppose in this first test tube we add the deoxydinucleotide triphosphate. Okay, we add thymine tri thymine trinucleotide phosphate. In second test tube we add dideoxycytosine triphosphate. In third slide we add dideoxyadenine triphosphate. In fourth test tube we add dideoxyguanine triphosphate. Okay, these dideoxy, thymine triphosphate, cytosine triphosphate, adenine triphosphate, guanine triphosphate does not contain the free OH group. That's why it terminate the amplification group. That's why this
DNDPs terminate the growing chain of growing chain of DNA. Okay, okay. Then we grow the complementary chain until the termination of termination die or termination step. Why in for the termination of growing chain, these dideoxynucleotide triphosphates are responsible. Okay. Then in next step, denature the growing chain. Then perform the electrophoresis separation of for the separation of DNA band according to their size. And by seeing the result of this agarose gel electrophoresis, we analyze the DNA sequence of the then we identify the DNA sequence of our desired target. Such first is a T, this one. Second is a C here. Then G. We read this gel electrophoresis step in this lower point. Then we make a complementary of these sequence, and finally we analyzed our result or sequence of the DNA. This is the Sanger sequencing method for the analysis the DNA sequence of our desired result. In Sanger sequencing method, first is the amplification method, second is the first is a denaturation step, second is in Sanger sequencing, first is a denaturation step, second is the amplification step, in third step we add the a primer in the test tubes, then we add the DNA primer, then we add the DNA polymerase, then DNDPs, then deoxydinucleotide triphosphate, which are responsible for the terminate the reactions. After performing the whole process, we separate the DNA sequence according to their size on a gel electrophoresis. By seeing the gel electrophoresis, we analyze our finally result. This is the Sanger sequencing method, and this is the first generation DNA sequencing method. Okay, now we discuss the second generation DNA sequencing method. First is the Roche 454 DNA sequencing method, Illumina Solexa DNA sequencing, AB solid DNA sequencing. These are the second generation sequencing method. Now we discuss the third generation DNA sequencing method. First is a Helicose Bioscience, Pacific Bioscience, and Oxford Nanopore. These are the third generation DNA sequencing method. First generation sequencing method is Sanger sequencing and Max Gilbert sequencing method. Second generation sequencing method is Roche 454 DNA sequencing method, Illumina Solsexa DNA sequencing method, AB solid DNA sequencing method. In third generation sequencing method, we discuss the Helicose Bioscience, Pacific Bioscience, and Oxford Nanopore. Okay.
Next, we discuss the computational biology.
Now we discuss the biological databases. First is a NAR databases. N stands for nucleic acid. N stands for nucleic. A stands for acid and R stands for research. Nucleic acid research database. Basically, this is a most extensive list of biological databases being maintained via International Journal International Journal Nucleic Acid Research, which publish a special issues for molecular biology databases. And the first issue is published in 1996. Next is a NCBI. NCBI stands for National Center for Biotechnology Information, and it is also maintained by the NCBI. The flash flow chart for 24 databases connected via line shows the relationship and interlink among all these databases. Okay, the databases are divided into major six groups, and these six groups are nucleic acid, nucleotide, protein structure, taxonomy, genomics, and expression. Okay, this NCBI also provide the link to individual database description page. Okay, next is the EBI. Next is the EBI database. EBI stands for European Bioinformatics Institute. Okay, the main portal of all EBI databases divided in several groups such as literature, microarray, nucleotide, protein, structure, pathway, and ontology. Okay, and it also linked to the database query and retrieval system can be found in this portal. Okay, this is a international journal. This is a NCBI. This is a molecular database. This is also a molecular database. NCBI stands for National Center for Bioinformatics Information. EBI stands for the European Bioinformatics Institute. Okay. Next is a genomics database. What is a genomics? Next is a genome database. Next is a genome database. What is a genome? Genome is a complete genetic material of an organism. If we want to extract the complete genetic information of an organism, then we use these database. That is a gold. G stand for genomes, OL stand for online, D stand for database. That is a genomes online database. It is a comprehension information resource for complete and ongoing genome sequencing project with the flowchart table of statistical data. Next is Carrier's genome. Carrier's genome is a collection of sequenced genome with brief description and reference for each genome. The direct link of the sequence database in EMBL or annotations in ENS EMBL make it very convenient. The direct link to the sequence database in EMBL or annotation in ENS EMBL make it very convenient for the user. Next is CropNet. CropNet is the website of UK crop plant informatics. crop plant bioinformatics, network to develop management and description of information related to comparative mapping and genome research in the crop plant. Okay. First genome database is GOLD. GOLD stands for Genome Online Database. With the help of this database, we easily extract the information of complete and ongoing genome project or sequencing project in a form of flowchart and statistical table. First genome database is a GOLD. GOLD stands for Genome Online Database. Okay, this Genome Online Database is a comprehension information. This database gives the comprehension information about the complete and ongoing genome sequencing project with flowchart and in the with the in this first is a GOLD. GOLD is a genome online database. This database gives the comprehension information about the complete and ongoing genome sequencing project and the information we extract the information of genome in a form of flowchart and statistical table. Okay, next is the Karen. Next is the Karen's genome. In Karen's genome is a collection of sequenced genome with brief description. Okay, what is the next sequence? Next database is a CropNet. CropNet is a website of UK crop plant bioinformatics. Okay, we all in this database we extract the information about the crop plants. Okay, this is the genome database. Now we discuss the sequence databases. In sequence databases, first we understand the genome sequence database. First database is the GenBank database. This GenBank database is the web portal to the NIH genetic sequence maintained by the NCBI. NIH stands for National Institute of Health. NCBI stands for National Center of Biotechnology Information. Okay, this GenBank is also a part of International Nucleotide Database collaboration. Okay, we literature citation, release note, and an example record can be found in this page. Next is a EMBL. EMBL is a web portal of EMBL nucleotide sequence database and it is maintained by the EVI, that is the European Bioinformatics Institute. Okay, it is also a part of International Nucleotide Databases collaboration. Various documentation such as release note, data statistics, user guide, feature table definition and sample entry is available in the home page of EMBL. Okay, next we discuss the next is a RefSeq. RefSeq stands for the reference sequence collection and it is constructed by the NCBI to provide a comprehensive, integrated, non-redundant set of DNA, RNA, sequence and protein products. Okay, it provides a stable reference for genomic annotation, genome genome annotation, gene identification, characterization, mutation, polymorphism analysis, expression study and comparative analysis. Next is a Unigene. Unigene is an organized view of transcriptome and this is created by the NCBI. Each unique gene entry is a set of transcript sequence that appear to come from the same transcript same transcription locus together with the information and protein similarities, gene expression, cDNA clone reagent, and genomic location. Okay, next is DPSNP, database of single nucleotide polymorphism. It is also maintained by the NCBI. Okay, next is the EMBL CDS. CDS are database of nucleotide sequence of coding sequence, and this is also maintained by the EMBL. Okay, first is a RefSeq. In RefSeq is a collection, and RefSeq is a reference sequence collection. It is also constructed by the NCBI. Okay, next is the Unigene. It is also maintained by the NCBI. Database of single nucleotide polymorphism. It is also maintained by the NCBI. EMBL CDS, it is maintained by the EMB. Now we discuss the protein sequence database. Swissport, the entry site for well annotated protein knowledge database maintained by the SIV at Geneva, Switzerland. The list of reference, comprehension, user manual, data, statistics with table and flowchart are provided. Okay, in Swissport, basically we extract the information of protein sequence. This is maintained by the SIV. Next is the Uniport. Uniport is the main website for International Protein Sequence Database, which consists of protein knowledge database, that is Uniprot KB, the sequence cluster, that is a Uniref, and sequence archives, that is a Unipatch. Next protein database is a Human Proteomics Initiative. And EMEBI project to annotate all known human sequence according to the quality standards of Uniprot KB and Swissport. Okay, it provides for each known protein a wealth of information that include the description of its work, its domain structure, subcellular location, post-transcriptional modification, variation, similarity, and similarities to other proteins. Next is a IPI. That is a the website IPI stands for the International Protein Index Database, which provide a top reveal guide to the main database that describe the proteomics of proteome protomes that describe the protomes of higher eukaryotic organisms. First protein database is a Swissport. It give a comprehension information about the protein sequence database. Second is a Uniport. Uniport is a main international protein sequence database. Next is a HPI. HPI stands for Human Proteomics Initiative. Next database is IHPI. HPI stands for Human Proteomics Initiative. Provide information for each protein, a wealth of information that include protein's description, its work, its domain structure, subcellular location, post-transcription modification, variation, similarity to other protein. Next is the IPI. IPI stands for International Protein Index Database, which provide a top reveal guide to the main database that describe the protein information, the whole protein information related to the higher eukaryotic organisms. Okay, next we discuss the protein domain database. First is a ProSite. ProSite is a database of protein domain, families and functional site, and this is created by the Swiss Institute of Informatics, Bioinformatics. Next is a print. Print is a database of protein fingerprint consisting of conserved motif within a protein family, created and maintained by the Manchester University, UK. Next is a blocks. Blocks is a database of multiple aligned ungapped segment corresponding to the most high conserved region of protein, created and maintained by the Fred Hutchinson Cancer Research Center, USA. Next is a CDD. CDD is the databases of conserved protein domain, created and maintained by the NCBI structure group. Next is a PRODOM. PRODOM are database comprehension, a set of protein domain family automatically generated from the Swissport and TREMBL sequence database developed and maintained by the University Cloud Werner France. Okay. Protein domain databases. First is a protein domain databases. First is a ProSite. ProSite is a database of protein domain family and functional site, and this ProSite is created by the Swiss Institute of Bioinformatics. Second is a prints. Prints is a database of print fingerprint consisting of the conserved motif within a protein family, and it is created by the master. This is created and maintained by the Manchester University, UK. Next is a blocks. Blocks is responsible for the databases of multiple aligned ungapped segment created to the motif highly conserved region of protein, and it is also created and maintained by the Fred Hutchinson Cancer Institute of USA. Next is a CDD. CDD is a conserved protein domain database. Next is a PRODOM. PRODOM is a comprehension set of protein domain family automatically and generated by the Swissport and TRMBL. Okay. Next is a protein family database, that is a PFAM. PFAM is a protein family. A database of protein families represented by the multiple sequence alignment and hidden Markov model constructed and maintained by the Sanger University, Sanger Institute, UK. Okay. Protein family database, that is a PFAM. PFAM is a database of protein family represented by the multiple sequence alignment and hidden Markov model construct, and this PFAM is constructed and maintained by the Sanger Institute, UK. Okay. Now we discuss the protein functional database. This is a protein domain database. This is a protein family database. This is a protein sequence database. And this is a protein functional database. Now we discuss the protein functional database. First is the IMGT. IMGT stands for the International Immunogenetic Information System. The International Immunogenetic Information System is a high quality integrated knowledge resource of immune system of human and other vertebrate species, and this international immunogenetic information system is created and maintained by the University of Montpellier, France. Okay, IMGT is a databases for immune system of human and other vertebrate species. Next is a HPA. HPA is a website for human protein atlas, which HPA stands for Human Protein Atlas, which show the expression and localization of protein in a large variety of normal human tissues, cancer cells, cell line with the aid of immunohistochemistry image development and maintained by the proteome Research Center, Sweden. Okay, protein functional databases. First is the IMGT, second is the HPA. IMGT is stand for International Immunogenetic System Information System, and IPA stands for Human Protein Atlas. IMGT is maintained and created by the University of Montpellier, France, and HPA is developed and maintained by the Proteome Research Center for Sweden. IMGT is responsible for the integrated knowledge resource of immune system of humans and other vertebrate, and HPA is responsible for showing the expression and localization of protein in a large variety of normal human tissues, cancer cells, cell line, etc. Okay, now we discuss the protein structural database. That is the first is a RSCB. RSCB is the main repository of macromolecular structure maintained by the Research Collaboration for Structural Bioinformatics. Next is the MMDB. MMDB, the macromolecular database maintained by the NCBI. Next is the MSD. MSD, the entry pointed for the EBI macromolecular structural data. Next is the MSD Lite. MSD Lite, the web server providing the simple search of protein structures. Okay. Next is PDB Sum. PDB Sum, the EBI web server which provide the overview, systemic diagram and interaction of structure. Next is a BMRB. BMRB is a biological magnet resonance data bank. It is maintained by the University of Wisconsin Medicine. Next is a Modbase. Modbase, the database of comparative protein structure model developed and maintained by the University of California, San Francisco. Okay, these are the protein structural databases. Okay, now we discuss the classification of protein structure. First is a scope, second is a cat. Scope, the database of structural classification of protein developed and maintained by the Cambridge University. Next is the CATH, the database of calcification, architecture, topography, homologous family, developed and maintained by the University of College London. Next, we discuss the visualization of protein sequence structure. That first is a GenaLib. GenaLib, the GENA library of biological macromolecule, which provide the information on macromolecule structure with an emphasis on visualization and analysis. Next, we discuss the list of bioinformatics tool at International Bioinformatics Center. First is the Expressi tool. This Expressi tool is a comprehensive list of online web-based bioinformatics tool provided by the Expressi and worldwide. Next is the EBI tool. EBI tool is the entry page for the EBI bioinformatics tool. Next is the NCBI tools. NCBI tools, the entry page for NCBI bioinformatics tool. Next we discuss the tool for pairwise sequence alignment. First is a align, second is a blast tool. Align, the web servers for pairwise sequence alignment maintained by the EBI. Okay, we also perform with the help of this align tool, we perform local sequence and local alignment as well as global alignment. Okay, next is the blast tool. Blast, the web servers for alignment of two sequence using the blast, and this blast tool is maintained by the NCBI. Okay, now we discuss the tool for multiple sequence alignment. First is the cluster omega. Cluster omega, the web service for global multiple sequence alignment maintained by the EBI. Okay. Second is a muscle. The web service for multiple sequence comprehension via local expectation maintained at EBI. It claimed to achieve better accuracy with high speed than cluster omega depending on the choice of objection. Next is a MAFAT. MAFAT, the web service of high speed multiple sequence alignment using fast Fourier transformation, and this is maintained by the EBI. Next is a Kaline, the web server of fast and accurate multiple sequence alignment. This is also maintained by the EBI. Next tool is Tcoffee. Tcoffee is the web service of several tools for computing and evolution and manipulation of multiple sequence alignment of DNA, protein sequence and structure. This tool is developed and maintained by the Centric Notre Dame at Center for Genomic Regulation, Spain. Sequence alignment tool is the Dieline, the web service for multiple sequence alignment. This is maintained and developed by the Burkhard Murgistron at Wilhelm University, Germany. It use it uses a local alignment approach to compare a whole segment of sequence without gap penalty. Okay, there are a lots of multiple sequence alignment tools are available. First is a cluster omega, second is a muscle, next is a muffard, next culling, then tea coffee, then Dieline. Okay, these tools are used to perform the multiple sequence alignment. Okay, now we discuss the tool used for gene identification. First is a GenScan. GenScan, the web service for identification of complete genome structure in genomic DNA, and this tool is maintained by the MIT. Okay, next is a GenID. GenID, the web service for prediction of gene in anonymous genomic sequence, developed and maintained by the University of Pompeu Fabra, Spain. Next is the HMM gene, the web service for prediction of vertebrate and C. elegans gene, developed and maintained by the Denmark Technical University. Next is a software, the web service for gene prediction, limited free uses for academic users. Okay, these are the gene identification tool. First is a GenScan, next GenID, next HHM gene, and then software. Okay, next we discuss the visualization and modeling tool. For the visualization tool, we use the Swiss PDV viewer. Next we use PyMol. Next 3NCD. Next RASMOL. These are the visualization and modeling tools. Okay. Next we discuss the phylogeny program. First is the PyList. PyList, the website for comprehension package of phylogeny interference package. This PyList stands for phylogeny interference package. This is created and this is maintained by the Geo Felixton at University of Washington. This package can be downloaded and installed on Linux, Windows and Mac freely. By the help of this program, we easily construct the phylogeny tree. Next is a tree puzzle. In tree puzzle, for the construction of phylogeny tree, in tree puzzle we use the maximum likelihood method. Next is a PAML. For the construction of phylogenetic tree, in this method we use the maximum likelihood method. Next tool is MEGA, the website for molecular evolutionary genetic analysis package, developed and maintained by the Masatoshi Negi and his colleagues. It is originally designed for the Windows platform with the graphical interface and uses the distance method to construct the phylogeny tree. Okay, These are the program by which we easily construct the phylogenetic tree and study the evolutionary relationship between the organisms. Now we discuss the types of biological database. First is a primary database. Second is a secondary database. Primary database consists the database derived from the experimentally, such as nucleotide and 3D structures. 3D structure of protein are the example of primary database. An example of primary database is GenBank, EMBL, DDBJ, and PDB. Next is a secondary database. Secondary database is derived from the analysis or treatment of primary database. Okay. The secondary sequence database contain the information like conserved sequence and active site residues of protein families arrived from multiple sequence alignment set of related protein. And the example of secondary database is translation EMBM and SCOP. Okay, these are the example of primary database. First is the EMBL, that is the European Molecular Biology Laboratory. Next is the DDBJ, DNA Data Bank of Japan. Next, Swissport, then PIR, Protein Information Resource, then Uniport. Okay. Secondary database is TREMBL, translation of EMBL nucleotide sequence. Then protein family, PFAM, then ProCyte. These are the example of secondary databases. These are the sequence databases. Now we discuss the structural databases. The example of primary structural database is PDB, that is a protein data bank. Second is a secondary structural databases. First is a SCOP, that is a structural classification of protein. Next is the CATH, class architecture topography and homology. Next, NDB, nucleic acid database. DSSB, database of secondary structural assignment. Next, HSSP, homology derived secondary structure of protein. These are the sequence database. This is the primary sequence database. This is the secondary sequence database. This is the primary structural database. This is the secondary structural databases. Now we discuss the functional and pathway databases. Genome databases we already discussed in our previous slides. First is the KEGG, that is the Kaito and next this is a Kaito Encyclopedia of Gene and Genomes. Okay, our data set for understanding the high level function of biological system such as pathway, molecular network. It includes the information on metabolic pathway, disease pathway, and gene function. Reactome, the created database of biological pathway and reactions. It provides the detailed information on pathway related to metabolism, signaling transaction, and disease process. Basically, this database is responsible for the biological pathways. Next is the GO, that is the gene ontology. The comprehensive resource for gene functional classification provide the standardization term for gene product attributes across the species, include the biological process, molecular function, and cellular component.
This is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.